So welcome everyone to the first week of our MOOC. We're hoping that you enjoyed it so far. Thank you so much for putting in comments about your experiences. We are loving every comment and we promise you that we do read them and we wanted to see how you're getting on. These are very important, particularly in areas where we can improve. For example, we have read that the little arrows that we had to talk you through histology in Professor Stefan Hupscher's video were not as clear as they could be. In new iterations of these videos, of course, we're going to make the curses bigger and more easy to follow. You also had some questions about the description of the histological state. So in this video, we're going to give you a little bit more detail about this histology in the normal and in the cirrhotic livers. We also wanted to give you an idea of what it is like to work in a laboratory, and Trish and I are both scientists and teachers, so what we're doing this week is we're preparing for our teaching sessions. We're also supervising students that are doing work in the lab, so we're going to introduce you to some of our students. My lab is interested in the role of immune cells in viral infection and chronic inflammation in the liver, so I'm going to introduce you to Ashley Retchless, who is a medical student and is undertaking research here and he's looking at viral hepatitis. I'm also going to introduce you to two of my PhD students, Sudha Purswani and Ben Wiggins. Ben has just started and Sudha is working on B cells and their role in chronic inflammation of the liver. And that inflammation can be of viral origin, autoimmunity, dietary injury. We are really relying on people donating liver for these experiments. We are looking at human liver samples that we collected from the adjacent Queen Elizabeth Hospital. So this video is also one of the greatest opportunities that we get to thank you for donating tissue. We know from your comments that quite a few of you are patients with liver diseases and some of you have already had transplants, so thank you very much for these. We hope you enjoy it. Keep the comments coming.
Some of you requested more detail on how we stained our liver specimens in the video with Stefan Hupscher, so we'll show you how here. Here, one of our pathologists, Melissa Wardale, is cutting a block of tissue which is prepared in wax into very thin sections. These are less than the thickness of a human hair. They're then floated on water in the black water bath you can see to the right, so that she can pick them up and individually place them onto microscope slides. The slides are then immersed in a staining solution so that you can see the tissue, because otherwise it's transparent because it's so thin. We used two stains in our interview with Professor Hupscher, hematoxylin and eosin, which is very widely used, and here the hematoxylin stains the nuclei blue, whereas the eosin stains the cellular cytoplasm a sort of pinky orange. And we also used a Van Giesen stain, and this is a differential stain for collagen, where the cells will appear a brownish yellow with a blacky nucleus, and the collagen is a very bright pinky red. The method is the same, you just use different baths, as you can see in the video here, to dip the cells into for the two different stains. Now we want to highlight the differences between normal and cirrhotic liver, when we've used either a H&E stain or a Van Giesen stain. And in the pictures which follow, we have normal or alcoholic liver tissue, and we've highlighted the key features for you. This is a Van Giesen stain of normal tissue, and you can see that there's very little pink stained collagen other than to provide structure and support around vessels in the portal area. You can also see the presence of steatosis in the centre of the section. This is now a section from a patient with alcoholic liver disease, and you can see much more inflammation in this tissue and structural disorganisation, but you actually need the Van Giesen stain to see the collagenous scar tissue very clearly. So now this is the same patient's tissue sample, so it's an alcoholic liver, but again you can see now with the Van Giesen stain it's much more obvious just how much of that red collagen material there is in this cirrhotic liver specimen.
So this is Ash Gretchless. He is an intercalating medical student that spent some time in the lab to do experiments. Could you tell us a little bit about what you're doing today?
Hi, yeah, sure. Today I'm setting up for one of my experiments that I'm doing today. Basically, I'm counting out white blood cells, which are immune cells, which we're going to use in an infection later to transfer a virus onto liver cells.
What virus are you using?
We're using hepatitis C virus. This is going to be done in the high safety laboratory where everything's kept under control.
Brilliant. So how are you going to measure infection after you have transmitted virus?
After we put the virus onto the liver cells, we're going to stain it with an antibody that makes it glow, and then we're going to take this into the dark room with the microscope, and I'm going to sit there for many hours counting and looking for little glowing liver cells.
Brilliant. So what's infected is going to be green, and the non-infected are going to be non-green.
Dark.
And you have to count numbers of infected cells. Oh, that's fun. So why are you doing this?
I'm doing this to hopefully develop a means of testing new medications that are used to treat viral hepatitis. Hopefully we can pass this on to drug companies to help them test out all the different drugs they come up with and hope that one of them will be more specific to patients.
This looks really interesting, generating antivirals coming through. Thank you very much. So this is Sudha Praswani and Ben Wiggins, PhD students in my lab. Working very hard, as you can see today. So Sudha, what are you up to?
Basically, we got a piece of normal liver tissue today from the QE, and I've just processed it and extracted the B cells from it.
So was that from explanted liver that was from an operation that happened this morning?
No, it was actually from a patient who underwent a laceration of the liver in an accident, so it's normal liver, and they had a transplant, so we got the explanted full donor liver.
That's quite rare, isn't it?
Yeah,
it's normal livers in the laboratory. So what's your experiment today?
Well, now that I've extracted the cells, I'm going to stain them for different B cell subsets. And this will be our control liver to compare to other disease livers that we've received in the past.
And Ben, you're helping out?
I'm learning and sort of helping out here, yeah.
He's my assistant.
I am, exactly.
Do you usually get a lot of B cells out of normal liver?
It depends. Some livers it can be 16 million and some can be 60. It just depends on the size of the tissue that we receive. And well, what we're trying to see is if their disease contributes to the number of B cells that there are. So in certain diseases you may get more. So I'm just going to count the cells now. 101. 101 divided by 2 times 40. 20 million. Don't know. Never mind.
Is that what you expected?
Well, it's better than what we usually get.
Okay.
But we still haven't been to it next door.
It's probably because you let me do it too.
And it's a lovely day at the QE today. This lab is right opposite the hospital. Have to check it out.